Hi, my name is Viviana Jessey.
Hello, my name is Natasha and Jayong.
Hello, my name is Foo Liong Lin.
Hi, my name is Narmathaa.
Two sets of three separate microcentrifuge tubes were prepared and 5 drops or a small amount of solid 3 to 4 granules of all the lipid-containing products that had been brought were added. Their appearance and smell were described in the table of results. For set 1 solubility in polar solvent, about 1 milliliter of water was added to each centrifuge tube. The tubes were kept and shaken to mix. Observations were recorded in the table of results. For set 2, solubility in the nonpolar solvent, 0.5 ml of toluene was added to each sample. The tubes were kept and shaken to mix. Observations were recorded in the table of results. In this experiment, we tested 3 lipid-based food samples, G, shortening, and olive oil. For appearance, G appeared pale yellow and oily. Shortening had a creamy white color with a smooth texture, and olive oil was a clear yellow liquid. For odor, G gave off a grassy and fruity aroma. Shortening had a fatty, waxy smell, while olive oil had a distinct grassy fragrance. Next, we tested the solubility of each sample. All 3 lipids were insoluble in water, which confirmed their hydrophobic nature. However, they were soluble in toluene, a nonpolar solvent, supporting the fact that lipids dissolve well in nonpolar environments. These results confirm the characteristic properties of lipid. They are nonpolar, oily substances that do not mix with water but do dissolve in organic solvents like toluene. In conclusion, this experiment successfully identified the physical properties of different lipids, G, shortening, and olive oil, based on their appearance, odor, and solubility. G appeared pale yellow with an oily texture and grassy fruity aroma. Shortening was creamy white with a smooth texture and had a fatty waxy smell. Olive oil was a clear yellow with a distinct grassy scent. All 3 lipids were found to be insoluble in water, a polar solvent. This confirms the hydrophobic nature of lipids and support the principle that like dissolve like. We also explored the relationship between lipid structure and physical state. G and shortening, which are rich in saturated fatty acids, were solid or semi-solid. In contrast, olive oil rich in unsaturated fatty acids remain liquid at room temperature. Lastly, we identify common food products that contain lipids such as coconut cream, butter spreads, mayonnaise, and peanut butter. These lipid-rich foods play an important role in energy storage, flavor enhancement, and nutrition in our daily diets.
I learned how to observe and compare the physical properties like appearance, odor, and solubility of different lipids. I also understood how lipid solubility relate to their polarity, hydrophobic, and hydrophilic nature. In addition, I also gained insight into how fatty acid structure influence lipid physical state. Lastly, I also improved skills in handling small volume lab equipment like microcentrifuge tubes and droppers.
Challenges faced. First, difficulty in distinguish subtle differences in odor among the lipid sample. Second, ensuring proper mixing and observation during solubility tests, especially for thicker samples.
Use digital tools such as odor intensity scale or color comparison chart to standardize appearance and odor observations. Extend the experiment to include other lipid types such as weight and phospholipid for broader comparison. Allocate more time for observations and discussion to deepen understanding.